Dear students, welcome to this lecture series in botany. Today our topic is Pteridophytes, general characters and classification. In this lecture, we are going to discuss first of all the general characters of Pteridophytes, then followed by a brief outline of Pteridophyte classification, and lastly, diagnostic features of different groups of Pteridophytes. As we know, like all other living organisms, plants are also believed to have originated from their aquatic ancestors. In this evolutionary transition from aquatic to terrestrial habitats, a group of plants, commonly known as vascular plants or sometimes Tracheophytes, developed an efficient conducting system which consists of xylem and phloem. The evolution of this conducting system solved, number one, the requirement of water, number second, the food transport throughout the plant body, which is an essential requirement for an organism to grow on terrestrial land. Also, in the vascular plants, the cell walls synthesize a complex polymer of phenolic compounds called lignin, provides rigidity to the plant body in order to reach great heights. Presently, a number of groups of plants are recognized, both living and extinct. Traditionally and more commonly, the three main groups of plants under the vascular plants are Pteridophytes, Gymnosperms, and Angiosperms. Pteridophytes, which is the topic of today's lecture, are the vascular plants which lack seeds. That means they are the land plants which do not produce seeds. Unlike bryophytes, which are non-vascular land plants, here in Pteridophytes, sporophyte is the dominant generation. The sporophytic plant body is well differentiated into a true root, stem and leaves, which provides a system well suited to the demands of life on terrestrial land. Pteridophytes generally occur in plains as well as in mountains. They occur in cold, moist and shady places. They also occur in humid and tropical climates and usually grow on soil, rocks, in ponds and as epiphytes on other plants. Many pteridophytes, especially ferns, rely on symbiotic association of the mycorrhizal fungi. Pteridophytes have several uses. Some of the pteridophytes, they are used as food. Many pteridophytes, in particular the ferns, they are used in horticulture industry. The floating mosquito fern, namely Azolla, is used as a biological fertilizer because it has the capacity to fix atmospheric nitrogen. Flip side of these pteridophytes is, in particular the ferns, some of these ferns, they can be noxious weeds. Coming to the first part of this lecture, that is the general characters of pteridophytes. In pteridophytes, the plant body comprises of two distinct phases. Number one, the diploid sporophyte, and number second, the haploid gametophyte. We will discuss first of all the diploid sporophyte. The diploid sporophyte is the dominant phase in the life cycle. This sporophyte is independent of gametophyte. The sporophyte, which I already mentioned that it is the diploid phase, is generally herbaceous and is differentiated into a true root. Roots are usually adventitious, stem and leaves. The leaves may be small, microphyllous, or they may be large, macrophyllous, usually called as fronds. The leaves are chlorophyllous and autotrophic. All vegetative parts of pteridophytes, possess vascular tissues, which are organized into a system what we call as vascular bundles, or a collective term is used, stele. So we can say the pteridophytes are the first vascular plants in the evolution of plant kingdom which have developed a vascular system. The sporophyte, diploid sporophyte, performs both vegetative and asexual reproduction. The vegetative propagation takes place by buds that develop on the rhizome, or it can be by fragmentation of the rhizome. Second, the asexual reproduction takes place by means of spores, which are produced inside the sporangia. The sporangia are born on lower surface or in the axils of fertile leaves, known as sporophylls. The sporangia are born either singly or in groups. These groups we call them as sori. In pteridophytes may be either homosporous. Homosporous is the condition when the plants produce only one type of spores, or they can be heterosporous when the plants produce two different types of spores, the smaller microspores and the larger megaspores. A sporophyte produces haploid spores by meiosis. The spores, which are haploid, in turn germinate into a haploid gametophyte. Now, by this we come to the end of first phase of the plant body, that is the sporophyte. The second phase is gametophyte, which is the haploid phase. The germination of haploid spores, it gives rise to a gametophyte. The gametophyte is a small, simple structure with a short life cycle and called as prothallus. Homospore species produce bisexual gametophytes. On the other side, the heterospore species produce unisexual gametophytes. Here, the microspores germinate into male gametophyte, whereas the megaspore germinates into female gametophyte. The gametophytes, they show a ventral and a dorsal differentiation. The gametophyte is usually photosynthetic except in heterosporous members. The gametophyte reproduces sexually, and the sexual reproduction here is oogamous type. Now, the male sex organs called antheridia produce sperms by the process of mitosis. The female sex organs called orchigonia produce egg by mitosis. Orchigonia are multicellular with sterile jacket but without a stalk. Fertilization, that is the fusion of male and female gamete spores, occurs in the presence of water and takes place in the venter of orchigonium. The diploid zygote so formed develops into an embryo in the orchigonial venter. The embryo now grows by means of mitosis into a sporophyte. This sporophyte will be a diploid phase. Therefore, we can say the life cycle in pteridophytes is diplohaplontic. It means that the life cycle shows a heteromorphic alternation of generations between, on one side, the diploid sporophyte and on the other side, the haploid gametophyte, which are independent of each other. Now, the second part of the lecture, that is an outline of the classification of pteridophytes. Several classification systems for pteridophytes have been produced from time to time. The most recent classification is that of Smith and his co-workers, very recently in the year 2006. The classification is based both morphological data as well as molecular data. At present, majority of the workers who work on pteridophytes, they recognize 7 main groups in the class Pteridophytes. These are number 1, Rhiniophytes, number 2, Lycophytes, 3rd, Ophioglossoid Ferns, 4th, Psilophytes, 5th, Equisetophytes, number 6th, Marital Ferns, and the last and the 7th, Pteridophytes. Now, let us discuss the diagnostic features of these 7 different groups one by one. Number first, Rhiniophytes. The Rhiniophytes, represented by only the extinct fossil plants, were among the first vascular plants. It includes the genus Rhynia, a well-known vascular plant from the early Devonian, about 410 to 360 million years ago. The sporophyte consisted of dichotomously branched axes that bore terminal sporangia. They lacked both roots and a leaf-bearing shoot system. These two features evolved later within the Lycophytes. The system of Rhiniophytes were protostelic, in which the protoxylem, that is, the first formed xylem, was at the center. Now the second group, Lycophytes. The Lycophytes are commonly known as lycopods, or a lineage of plants that diverged after the Rhiniophytes. The roots of Lycophytes have an end-arch protoxylem. The stems have an X-arch protoxylem just reverse of the roots. Lycophytes have a sporophyte leaf structural type known as lycophyll. These are characterized by absence of a gap in the stem vasculature and have a single unbranched vein. The Lycophytes at present comprise of several extinct genera, for example, Lepidodendron and others. The only Lycophyte that survived to the present are small herbaceous plants, usually grouped into 3 main families: Lycopodiaceae, Selaginellacea, and Isoetaceae. First, the Lycopodiaceae. The Lycopodiaceae, which are often called as culp mosses, are distinguished in having one type of spore, that is Humosporus. It contains about 300 species which are distributed among 8 genera. Although resembling mosses, they are true vascular plants, wherein the long-lived plants is the sporophytic phase. Sporangia of the Lycopodiaceae develop laterally in the axil of the specialized leaves called sporophylls. The sporophylls differ in size and shape from the vegetative leaves and are aggregated into a terminal shoot system. This determinate reproductive shoot, consisting of a terminal aggregate of sporophylls with associated sporangia, is known as a strobilus or simply a coon. The 2 other families in the Lycophytes are Selaginellacea and Isoetaceae. These 2 families, they differ from Lycopodiaceae in being heterosporous. These also differ in having a characteristic flap of tissue called ligule, which is located on the upper surface of leaves near the leaf base. Heterospory, as seen in these 2 families, refers to the production of 2 types of spores, microspores and megaspores. The gametophytes of Selaginella and Isoetes are endosporic. Endosporic means the gametophytes develop entirely within the original spore wall. The Selaginellaceae contain about 700 species in the single genus Selaginella, commonly called spike mosses. The second family, Isoetaceae, consists of about 150 species in the single genus Isoetes, commonly called Colvert or Merlin's cross. The plants consist of a comb-like system that has roots attached below and numerous needle-like leaves above. These are small herbs with prostrate rhizomes that bear true roots and upright shoots and lack secondary growth. The leaves are microphyllous and spirally arranged on their shoots. The sporangia are arranged in cones or along the shoots. Now the third group, Ophioglossoid Ferns. The Ophioglossoid Ferns are unique in that each leaf consists of a sterile segment which contains the photosynthetic blade or lamina and a fertile segment. The most common genera of the Ophioglossoid ferns are Ophioglossum, commonly called adder's tongue, and Botrychium, commonly called crab fern. Whereas the species of Ophioglossum have a simple undivided lamina and an unbranched fertile segment, those of Botrychium have a compound lamina and a branched segment. The roots are unusual in lacking both root branches and root hairs. The gametophytes here are non-photosynthetic, subterranean, and may contain mycorrhizal fungi. Now the fourth group, Psilophytes. The Psilophytes, commonly called as whisk ferns, consist of only two living genera, Psilotum and Mesipteris. Psilotum occurs in tropical and subtropical regions of the world, but Mesipteris is restricted in its distribution, mostly in Australia and South Pacific islands. Psilophytes have an independent, dominant, and free-living sporophyte. The haploid gametophyte here is small, subterranean, free-living, and may contain mycorrhizal fungi. The sporophyte consists of a horizontal rhizome that gives rise to aerial, photosynthetic, generally dichotomously branching stems. All the Psilophytes are homosporous, that is, they produce same types of spores. They lack true roots, only absorptive rhizoids arise from the rhizome. The leaves of Psilophytes are highly reduced, peg-like structures and may lack a vascular strand. In this case, they are termed as enations. The sporangia are 2 or 3-lobed, which is called as synangium. The synangia are yellowish at maturity and are subtended by a forked appendage. Now the fifth group, Equisetophytes. The Equisetophytes, also called as phenophytes, contain several genera of extinct plants and at present represented by a single living genera of Equisetum with about 15 species. These are homosporous herbs without a secondary growth. The leaves here are small, megaphyllous, with a single trace of vascular tissue. Today, the only living Equisetophyte or the species of Equisetum, commonly called as horsetails. These Equisetophytes have an extensive underground rhizome with adventitious roots. The rhizome gives rise to erect, aerial shoots. Here, the stem is ribbed. This ribbed stem contains epidermal cells that are impregnated with silica. The stems are hollow inside and the leaves are whorled. The leaves are laterally fused to form a sheath-like structure at the nodes. At the tip of some of the aerial stems are strobili or simply cones which contain sporangia. The spores are unique among the vascular plants in containing chloroplasts, an unusual appendage called elaters. The elaters are hygroscopic and uncurl the spore body upon drying. Thus, they aid in spore dispersal. The gametophyte in this group is small, about 1 mm across, green mass of parenchyma with absence of vascular tissue. Now the sixth group, Maratoid ferns. These are a group of 6 genera which were previously grouped with Pteridophytes, the true ferns. However, unlike true ferns, that is Leptosporangiate ferns, the sporangia are here Eusporangiate. They have large pinnate or bipinnate leaves with circinate vernation. The sporangia in this group are located on the lower surface of the leaflet blades. However, in some cases, the sporangia are fused into a common structure called synangium. Here, the gametophyte is photosynthetic. Last and important group is Pteridophytes, or what we commonly call them as ferns. The Pteridophytes, also known as felicales, polypodiales, or some call them as pteridales, refer to a group what we commonly call them as Leptosporangiate Ferns. The group contains the greatest diversity, with about 11,000 living species at present. These pteridophytes occur in almost every habitat, be that terrestrial or aquatic. The majority occurs in tropics, but some are found in the temperate mountain regions. All of these pteridophytes are perennial and herbaceous. Although some of these pteridophytes, they attain the size of small trees up to 100 meters of height, what we commonly call them as tree ferns. However, they never have secondary growth. The sporophyte in pteridophytes consists of a single axis, either an upright shoot or a prostrate rhizome that bears both true roots and megaphyllous leaves. The leaves contain vascular tissue and the veins are dichotomously branched. The leaves, also called as fronds, or compound ones with a central rachis and the lateral leaflets. These are of various shapes and sizes, ranging from less than 1 centimeter long in Azolla microphylla to several meters long in giant tree ferns. Initially, the fronds are in coiled form and expand by unrolling from the base to the tip, with new growth produced at the apex. In addition to photosynthesis, these leaves or fronds also serve as sporophylls. On the underside of the sporophylls are sore, which are formed by the clusters of sporangia. On the outer rim of sporangium is a single row of specialized cells collectively known as annulus. This annulus helps in spore dispersal. The sore may or may not be covered by a flap of tissue called indusium. Here in pterophytes, the gametophyte is small, heart-shaped structure covered with rhizoids on the lower surface but without a vascular tissue. The antheridia are born among the rhizoids and the orchigonia occur close to the gametophytic apex. All the aquatic ferns are heterosporous. The reproductive structures here are complex and generally organized into a spherical structure called sporocarps. The sporocarps allow the sporangia and spores to remain dormant for long periods of time, an adaptation which enables them to survive when the seasonally ponds dry up. So dear students, this was all for today's lecture. Hope you have understood the topic. We will meet again with a new topic. Till then, goodbye.